So, designing experiments so that we can learn what we intend is harder than it might seem at first thought. Qingwu Meng is an expert at conducting research with plants grown under different light qualities. Dr. Meng, what light sources do you use to generate various light quality treatments?
I use light-emitting diodes, or LEDs. These are semiconductor light sources that are very energy efficient and long-lasting. These are single-colored or monochromatic LEDs that can provide a specific color that we need, such as blue, green, red, or far red, which is just beyond the visible spectrum that we can see. We can combine different colors of LEDs to generate unique light quality treatments. In my experiments, I used research LED fixtures that had 6 different color channels. I used computer software to control how bright each color channel was. And this allowed me to create different light combinations for different experiments.
Very cool. So, you do your research in an indoor laboratory. Why don't you use a greenhouse?
Controlled environment agriculture is growing food crops in indoor farms or greenhouses, where we can monitor and adjust environmental factors to create an ideal environment for plants. We have better control of the environment indoors than in a greenhouse. There is sunlight in the greenhouse, which can change throughout the day and season. Part of my research focuses on indoor crop production, where we grow plants without sunlight. In this case, LEDs are the only light source for plant growth and development. So plant growth is more consistent indoors than in a greenhouse, which has changing sunlight over time. The indoor lab simulates an indoor vertical farm, so the results we generate in the indoor lab are directly relevant to commercial indoor vertical farmers. In addition, without sunlight, we can study how plants respond to changes in light quality very precisely, so we can expand our knowledge in photobiology.
Ah, yeah, makes sense. So, what are other important considerations for researchers to ensure that experiments with light quality provide meaningful, reliable results?
So if light quality is the only variable we're studying, it's very important to keep everything else the same. By everything else, I'm referring to the way we grow plants, the environment, and other light parameters. For example, across different light quality treatments, we need to sow seeds at the same time. We need to irrigate and fertilize plants the same way. We need to have the same temperature, humidity, and CO2 concentration. We need to have the same total light intensity and duration. By keeping everything but the variable the same, we can eliminate potential confounding factors that can affect the results. For example, if temperature is not uniform under different light quality treatments, any difference in plant growth may also be due to temperature instead of light quality alone. It's also important to set up light quality treatments correctly with a light sensor called spectroradiometer. We need to make sure light is uniform at the plant level. And when it's time to harvest plants, we need to use the same standards to collect plant data consistently. And finally, we need to replicate the experiments and randomize treatment locations from replication to replication, so we can make sure that we can reproduce our results.
Yeah, makes sense. So, it's not as easy as it might seem at first thought. Thank you so much for sharing your experience and expertise with us.
You're welcome. Thanks, Dr. Williams.